And our next speaker is Maria Davila from the Institute of Structural Biology in Grenoble. And she's going to tell us about the B12-dependent photoreceptor CarH, looking at it using time-resolved electron cryo-microscopy.
Can you hear me well? Okay. So hello, my name is Maria. I'm a third-year PhD student at IBS in Grenoble, and I'm going to talk a bit about what you said, using time-resolved cryo-EM to study a B12-dependent photoreceptor called CarH. So I wanted to start with a reminder that biomolecules in general are dynamic systems. They move a lot, and their movements cover a very wide range of timescales in time, and also of orders of magnitude in space. And this is something that should definitely be taken into account when you're studying your favorite biological systems. And let's say you're a biologist, you have some kind of system that you like, let's say Rubisco or something like that, and you want to know how it does, what it does, how it works. And for this, usually what you want is to know what's an intermediate state of it. And if you're a structural biologist, you want to determine a structure of this intermediate state. And there's different ways to do it. You could use some clever biochemistry, but what about doing it in real time if you were able to do it? So how you go about this is that you start with your system and you apply some kind of trigger to start your reaction. This trigger can be anything, mixing with something, light, temperature jump, magnetic field, I don't know. And then you let the reaction proceed for a certain time delay, which has to be precisely controlled. And after this time delay, you either capture some kind of information about the system, which is the case, for example, in time-resolved crystallography, or you freeze trap the system, so you quickly cool it down, which is the case in time-resolved cryo-EM, in what we call time-resolved cryo-EM. And then with this cryo-trapped sample, you go and characterize it, in our case, inside a transmission electron microscope, and hopefully get the structure of your reaction intermediate to better understand how your system is working. So now that you're a bit more familiar with the idea, in which time-resolved cryo-EM doesn't mean time-resolved microscopy, but sample preparation, so the big thing is to have a precisely controlled time delay between your trigger and the stop of the reaction, which for cryo-EM grids is vitrification. And this can be done manually for long time delays of, let's say, more than a few seconds in a conventional sample preparation machine. But if you want to go to less than 1 second, you need some kind of special experimental setup, such as the one that was presented this morning. And there's a lot of these that have been published over the last years. And usually you can kind of classify them depending on what the trigger is, which is either like a chemical trigger, which is mixing with another reagent or another protein, something like that. Illumination, which can be used to start reactions of photosensitive samples, but also to use caged compounds like caged protons, caged ATP, or something like that. And there's also some kind of special technique, which is the melting and revitrification developed at EPFL. And this allows to go to shorter time delays. So now that you're a bit more familiar with time-resolved cryo-EM, I wanted to show an example, in which we apply this technique in complementarity with time-resolved crystallography, and this is from my PhD project working on the protein CarH. So CarH is a bacterial transcription regulator. It controls the biosynthesis of carotenoids in response to light. How it works is that in the dark, CarH is a tetramer of 4 monomers, which are identical, and each monomer has a ligand, which is light sensitive, and it's a form of vitamin B12 called adenosylcobalamin. So it has this cobalamin part with a cobalt and an adenosyl group, which is bound by a covalent bond between a cobalt and a carbon. And this tetramer binds to DNA, it binds to the carotenoid promoter, and doesn't allow the rest of the genes that are necessary for the biosynthesis of carotenoid to be transcribed in the dark. Once it's exposed to light, this bond between the cobalt and the carbon breaks, and this is kind of a trigger. And this bond breaking triggers a series of structural changes that go from this very small change to a full dissociation of the protein into 4 monomers that do not bind DNA anymore. And so what we wanted to know is how does this go from such a small change of bond breaking to full dissociation? And that had been the work in the lab for the past few years. And from time-resolved crystallography, they were able to obtain the structures of some reaction intermediates at 10 nanoseconds, 3 microseconds, and 10 milliseconds, which all have small changes in the chromophore site. But the thing is, after 10 milliseconds, they could not obtain any more high-resolution structural information because the crystals stopped diffracting. And we think this is because the change in shape of the protein is so large that it disrupts the crystal packing. So we are crystallographically blind after this point. But we knew from time-resolved solution scattering that the time scale for dissociation was in the hundreds of milliseconds. So we wonder if you could learn anything more about the structure in this time range. And for this, we need a technique that allows us to have high-resolution structures without the need for crystals. And so we tried time-resolved cryo-EM. So first, we checked that CarH is actually a good sample for normal cryo-EM. So we determined dark state structures of both a truncated version of the protein, which is a sample used for crystallography. So it only contains the cobalamin, the cofactor binding domains, and also the full length structure of CarH in complex with a DNA fragment. We then checked that we can dissociate the protein on a cryo-EM grid, that we can actually start the reaction on this sample support. And when you prepare grids in the dark, you can see these particles, which are tetramers. However, when you prepare the same grids in which you illuminate the sample after blotting and wait for a few seconds for dissociation to happen, and then you plunge freeze, you just see like very tiny particles in the background, and these are monomers, which are 20, 30 kilodaltons, so they're very small. So once we knew we could do this, we switched to real-time resolved sample preparation, which we did at TU Delft in the laboratory of Arjen Jacobi, where they have a custom plunger that can use light activation. So here you can see the sample preparation process. This is the sample loaded onto a grid on the tip of a tweezer. And then there will be blotting to remove the excess liquid. And once you have this very thin film of protein, the trigger will be a LED flash. And after the flash, there will be a small time delay and then plunging. So there should be illumination, time delay, plunging. Of course, this is slowed down. We did shorter time delays. And also we worked in the dark because CarH is an extremely sensitive protein and it is an irreversible reaction. So once it has seen the light, you cannot work with it anymore. So we did this sample preparation. We tried different time delays, always working with a relatively long light pulse because we had an LED and not a laser. And something that has to be taken into account when you are doing light activation cryo-EM like this is that a very thin film, you need a huge amount of photons to photoactivate everything that's in there because this thin film of solution is not 100% chromophores. This is mostly empty space, so most of the photons go through without doing anything. So we had to work with long light pulses because we didn't have a laser at the moment. So I will show some of the results from this. We focus on a 300 millisecond dataset. We started with the easy part, which was picking tetrameric particles and getting an average reconstruction, which looked pretty good. However, the density around the chromophore site was a bit ambiguous. We knew this was a reaction intermediate because we don't have any more density for the adenosyl group that leaves after this cobalt carbon bond that breaks. And of course, we had still a tetramer, so it's not the final state of the reaction. But I could fit at least two different conformations of the core ring, so the big ring in the cobalamin part. And we tried a symmetry expansion and classification strategy on this. So the idea, it was a bit tricky because the shape of this protein makes that it's a dimer of dimers in which the C2 axes are not properly aligned. So you have to do two symmetry expansions. But we got an average reconstruction of one monomer and then with the 3D classification around the chromophore region. And we could separate two classes, which I'm comparing here in gray to the dark state and to the final light state. So the first one of those, the only change with respect to the dark state is that the adenosyl group has left. And then the second class, after the adenosyl group has left, we have a shift of the core ring of a few angstroms, 4 or 5, and binding to another histidine of the protein. And then the next step that needs to happen to get to the light state is binding to this other histidine, which was bound in the beginning to the core ring. And this state has been observed before. It's actually the endpoint in the crystallographic experiments. But this one, we thought we never saw it before. But actually, when looking back at some crystallographic data, we realized that we had already seen it in both an uncontrolled experiment performed at ID29 at ESRF and an X-fold experiment. The thing is that it was not observed in the fastest monomer. What I mean by this is that when we do time-resolved crystallography on CarH, which I remind is a tetramer, each monomer reacts at a slightly different speed, which means at a certain time point, you find different reaction intermediates in different monomers of the protein. And we usually look just at the fastest one, and this was one of the slower ones. Continuing with this problem of heterogeneity in the crystal structures, we had the question if that could come only from crystal packing, or if it was something of the protein that it had, I don't know, some kind of cooperativity or something like that. And from our data, we think now that we find a mixture of intermediates. These two intermediates, we can find them by retracing back where the particles, the symmetry-expanded particles come from. We can get particles from the two classes in the same original tetramer. So we have a mixture of them. Of course, this is not easy to say because there's, for example, some views of the particle in which you cannot distinguish easily between one and the other class because it's just a shift of 5 angstrom of a part of the structure. So if you see it, I don't know, from one side, it looks more or less the same. But there's still, we think, a mixture of those. And we don't think this mixture comes from the illumination conditions because we're using a quite long pulse, but we know that the time that it takes to form this intermediate is longer than that from spectroscopy. So we think we'd really have a mixture of intermediate states in the same tetramer, which is interesting to understand this protein. Also, so far, what I presented was only with the tetrameric particles, which are the easy to pick, easy to work with. But there's other things in the data sets that are heterogeneous. And there's some suspicious classes that look like trimers or dimers, and we would like to explore those, but it's very challenging and because we cannot get any 3D reconstruction. So that's a work in progress. And of course, we would like to explore some of these heterogeneity processing algorithms that are being developed by many people. So to summarize for CarH, we got high resolution dark state structures of the truncated and full length protein in complex with DNA. We did successful time-resolved cryo-EM sample prep experiments in Delft. We obtained 3D reconstructions of those and could classify different intermediate states at the monomer level in tetrameric particles of CarH, and we identified a previously unknown or not studied reaction intermediate of this protein. And our goal at IBS now is to set up time-resolved cryo-EM to be complementary to our time-resolved crystallography expertise, especially on light-sensitive proteins, but not only, because we can work with caged compounds. And for this, I am copying and adapting the experimental setup that we used in Delft. And yeah, I'm doing the first test at the moment, and it's starting to work like a plunger. So I'm hopeful for it. It's maybe not as beautiful as the one shown this morning. It's an interesting project. I guess you will see the light flash at some point. It's also a mixture of high-tech and low-tech technology, which with a very fast and precisely controlled plunger motor and then 3D printed parts. Yeah. And yeah, so with that, I will finish. I would like to thank everyone who helped with the experiments in this project, my supervisors, the organizers for selecting this talk, and all of you for your attention. If you have questions, I will try my best to answer. Thank you.